Hello, everyone. My name is Jane Siwek. I'm just wrapping up my first year as a PhD student in Dr. Jishnu Das's lab, and I'm excited to talk to you about a project we've been working on in the lab titled Sliding Window Interaction Grammar, which is a generalized interaction language model for protein and peptide interactions. So protein language models, as I'm sure a lot of you have heard, have exploded in popularity recently, and they're often employed for predicting protein-protein interactions. However, our current protein language models do not take interactions into consideration. So when protein language models are looking at interactions, there's two main embedding methods that they use. So oftentimes these models will embed these blue and red protein sequences separately and then concatenate the embeddings post hoc, or they will concatenate the sequences and then embed that concatenated product. And then they'll use those embeddings to predict interaction. But our concern is that proteins often work together with other proteins, and these interactions are essential to their functions. So we were wondering if there was any information being lost when not taking interactions into account. So that led us to wonder, can we develop a representation of an interaction between two protein sequences? So we developed an interaction language model to analyze these paired sequences. So let's say we have a protein-protein interaction between the blue and the red protein sequences here. The first step that we need to do is generate a vocabulary of the interactions by codifying the sequences into some numerical representation. So when we were considering these different representations, a simplistic solution could just be using some random encoding, but we wanted to do something a little more biochemically relevant, so we used a polarity or hydrophobicity-based score to get biochemically derived scores. So once we have these scores, we'll treat the document, the interaction like a document, and then embed that interaction using a language model. So in a more in-depth explanation of the language generation, the first step that we need to do is identify a target sequence and a window on an interactor sequence. So here I'll talk to you about an example of a receptor-peptide interaction, but I want to emphasize that you can use any two protein sequences, and I'll show you another example later where we do that. So here we'll identify our peptide as our window of interest, and then the receptor as our target protein sequence. So the next step is we'll calculate the difference in biochemical score between those amino acid pairs. So we'll take that peptide and line it up to the beginning of the target protein sequence, and we'll encode each of the sequences with that biochemical score, such as polarity or hydrophobicity-based scores. And then at each position on the peptide sequence, we'll calculate the change in biochemical metric at each of those aligned residues to get delta biochemical scores. And then once we reach the end of that peptide sequence, we'll take the peptide and slide it down one position on the target protein sequence and continue calculating these delta biochemical scores until we have slid that peptide down the entire target protein sequence. So now that we have these delta biochemical scores, we'll break the scores into k-mer-sized subsequences. So in a language context, you can think if the interaction is like a document, these individual scores are like letters, and then the k-mers are like the words in that document. And so the final result of the language generation is a set of k-mers that represents each of the interactions. So now that we have this interaction sequence-based vocabulary, we'll use these k-mers as an input into a doc2vec model to create vector representations of the interactions. And then we'll use those vectors to train a classifier and predict whether the proteins are interacting or not interacting. So overall, this is not a large language model, but a smaller and more biologically inspired interaction language model. So how well does this language really represent interaction? So we wanted to look into two difficult enough problems. And the first problem we landed on are peptide MHC interactions because it is a very well-studied problem, but we wanted to see how well can our language generalize. So peptide MHC interactions are really essential to our T-cell immune response because they bind to peptides and antigens and then present them to T cells for recognition by their receptors. But importantly, different MHC alleles will have distinct peptide repertoires that they present. And they also have two distinct classes. There's class 1 and class 2 MHCs, which are very sterically and structurally different from each other. Class 1 is capped on one end and binds shorter peptides, while class 2 is open on both ends and binds longer peptides. And again, this is a really well-studied problem of peptide MHC binding, and there's several methods already developed for this task, and these current methods train on as much data as possible, and they identify the binding motifs of the MHCs directly, and they predict really well on receptors that are close to their training set, but they don't do as well on receptors that are more different from their training set. So we wanted to see if our interaction language model could better predict these peptide MHC interactions. So we used immunopeptidome data from MS solution experiments, and we took 36 publicly available data sets and clustered them by their functionality. And so then we took just 8 of those most functionally different HLA alleles to build our training set. We also created a validation set of both proximal and distant HLAs, which you can see are pink and blue in the tree. And then we did a standard cross-validation and plotted the results in an ROC curve. So for anyone unfamiliar with an ROC curve, the x-axis is false positive rate, the y-axis is true positive rate. A perfect classifier has an AUC of 0.5, which will be that diagonal gray line. And a perfect classifier has an AUC of 1, which you can see in the green line. So our blue line, or a validation line, you can see we observed good signal, meaning we were able to predict which of these class 1 alleles bound which peptides. But what we really wanted to know is how well does swing do on receptors that are not in our training set? And as I said before, we chose some proximal class 1 HLAs not in the training set and tested our model on it. And as you can see, we perform really well, even on these unseen alleles. And then also to take it a step further, we also looked at those distant HLA alleles, and we're still able to observe signal, which was really promising. We also did a very similar setup with class 2 HLAs. We took 5 class 2 HLAs as our training set, and then you can see the validation set in blue on the tree. And we performed those similar experiments as before with the standard cross-validation and cross-prediction on the unseen data sets. And we were really excited to see that this class 2 model performed really, really well, meaning we were able to predict which alleles bound which peptides. And it was also much better than the class 1 model. So we wanted to ask, where is this information coming from? Because we're not encoding that motif directly, unlike the other methods. We wanted to ask, are we learning anything biologically meaningful here? So the class 2 motif is 9 amino acids long with a few flanking amino acids that are important for binding. And as you can see on the left, our training set distribution has peptide sizes 13 to 21. So we were curious what would happen if we started to do a deletion experiment and started shrinking those peptides by 1 amino acid at a time, and then specifically at what point do we start to lose signal? So that's exactly what we did. We deleted one amino acid of those peptides at a time, and you can see our performance drops off right around 8 amino acids, which lines up really well with our biological expectations because that means we had to remove at least one of those core amino acids from the motif. And so this experiment told us that we were in fact learning motif information without explicitly encoding it, and Swing was able to learn de novo structure of the class 2 motif. And then we wanted to ask can we learn about all motifs in this model regardless of class? And specifically, we wanted to ask if we learn on just class 1 receptors, can we predict on class 2 receptors? And like I said before, this is a really difficult problem because class 1 and class 2 HLAs are really, really different from each other. So they bind different peptides, they have different steric and structural properties, and they even participate in different downstream signaling. So it was a bit of a crazy idea, but we still decided to do it, and the answer was yes. So this is a model trained on the 8 class 1 alleles and predicting on class 2 receptors, which makes Swing, to our knowledge, the first model to ever be able to cross-predict across these classes. So quickly looking at some of the gold standard peptide MHC models, on the right side of the plot are the class 2 models, net MHC pan 2 in blue, mixed MHC 2 pred in orange, and then swing class 2 in magenta. And you can see that these class 2 models all performed very similarly, one predicting on the class 2 receptors, which makes sense. But when you look on the other side of the plot to the class 1 models, you can see that swing is the only model that was able to accurately predict on the class 2 receptors. And this emphasized to us that there was something about our interaction language that was transferable across these classes. But then we thought if our model was truly generalizable, we should be able to mix class 1 and class 2 data together, and the model should still be able to work. And this has never been done before because the classes are so different. But if we are truly learning something generalizable, we should be able to mix these data sets. So we created a mixed class model of 8 class 1 alleles and 2 class 2 alleles that also remained predictive. But then we wanted to give ourselves an even more difficult task, which was, can we predict class 1 to class 2 in another species, human to mouse? And to emphasize here, we're training on class 1 human data and then predicting on class 2 mouse data. So we looked into de novo class 2 data from 2 disease murine models. So in the SLE model, we're looking to identify the binders of the H2IEK allele. And then in the type 1 diabetes model, we wanted to identify the binders of the IgG7 allele. And so here we have swing class 1, class 2, and a mixed class model in blue, orange, and magenta, and then mixed MHC2 pred in green and net MHC2 pan in gray for comparison. And overall, we can see that we have much better accuracy than these other models when trying to identify these binders, while also being the only model that has never trained on any mouse data before. And then on top of that, you can see that we can predict beyond the typical length of peptides, which shows that swing doesn't have any length bias because we're not encoding that motif directly like the other methods are. And then furthermore, you can see that our models, irrespective of our classes, so the class 1, class 2, and our mixed class model, all perform very well, which emphasized to us that our interaction language was transferable. So overall, swing was able to do zero-shot learning in 2 ways, both in the prediction of de novo peptide MHC binding across organisms, human to mouse, but also classes class 1 to class 2. But then we wanted to give ourselves another really hard problem, and we wanted to see if we just change one amino acid on a protein-protein interaction, can we disrupt whether that mutation would disrupt the interaction or not? So we shifted our gears to the missense mutation perturbation context. So there's several state-of-the-art models in this field of variant effect prediction, such as alpha missense, which is an AlphaFold-based model. There's Eve that uses evolutionary coupling data for multiple sequence alignments. And there's ISMB, which is Meta's variant effect prediction model based off of the ESM-fold model. And notably, all of these methods use some sort of alignment or structural information that helps aid their prediction. But one important caveat of these models is that our modern variant effect prediction models learn pathogenicity, but not necessarily specific interaction disruption. So I could ask this model, is this mutation pathogenic or not, but not necessarily, is this specific interaction disrupted? So now we've modified our framework so that our sliding window is no longer that binding peptide, but it is a window around a mutation on a protein-protein interaction. And to emphasize again, these variant effect prediction models are very, very good predictors of pathogenicity, but not necessarily for specific protein-protein interaction disruption. So we wanted to test swing on different classes of missense mutations and specifically their interactions with experimentally validated Mendelian disease-associated variants and population variants. So we ran our standard cross prediction like before on each of these models for each of the types of mutations. And first you can see Mendelian disease-associated variants, which it's succeeded very well. We also tested population variants, which performed well too. And then we also created a mixed class model of both the Mendelian and population variants. So you can see that overall, Swing was able to identify which of these missense mutations disrupted specific interactions and which ones didn't, just from sequence information alone. But for a more stringent test, we wanted to ensure that Swing was learning relationships beyond just sequence similarity alone. So we clustered our interactions based on the sequence to group them by similarity. And then in a sort of nested fashion, we performed a proximity dropout experiment where we dropped the two closest clusters to the test set from our training data. So you can see that this model performed a little bit worse than our mixed model from before, but there's still a really strong signal despite these drop clusters, which showed to us that Swing was learning relationships beyond just similarity alone. We also benchmarked Swing against those variant effect prediction models that I mentioned earlier, and Swing was able to outperform all of these other methods on the specific interaction disruption task. So, in conclusion, Swing is a first-in-class interaction language model that learns the language of peptide-protein interactions across contexts using only sequence information. It's a zero-shot model that accurately predicted peptide MHC binding across HLA classes and species and was able to make accurate de novo predictions. It also learned biologically meaningful information without any prior knowledge and was able to predict the impact of missense variants on specific interaction disruption or specific protein interactions using sequence alone, which no variant effect prediction method has been able to do before. So here's the links to our preprint and our GitHub. We're currently under revision, so we would love to hear any suggestions that you might have. I'll leave it up another second. Yeah.
Thanks very much, Jane. So I think maybe one question, but I really think we should. Okay.
I think great talk, by the way. Really nice work. I think you partially answered what I was going to ask, which was about it seems like this is beyond everything, like species, classes, and all of that. And it seemed like it's all about sequence similarity. Mm-hmm. So did you go back and all the first results that you have presented looked at the sequence similarity type of baseline and then see where they are? Because that's what I've been wondering throughout. Yeah. And then it came a little late.
So I think we definitely did it for the mutation ones, and we're still trying to figure out better methods for like figuring out like a baseline of sequence similarity. But I think, for the peptide MHC, I believe we're also gonna look into that as well. But
Thank you.
Thank you.